Warburg thought that glycolysis was impaired and got into long arguments with a biochemist at who was, I think, Fell's Research Institute, Sidney Winehouse. And after a while, no one cared who read their arguments, because what could you do with it anyways? And now the truth is that glycolysis is speeded up. It's not a failure of respiration. That is, the cancer-inducing genes, one way they induce cancer is they turn up glycolysis. And my interest in cancer was such that in the first edition of the Molecular Biology of the Gene, I had a chapter on cancer. So, because I had I gave a course on cancer in 1959. So it's been a big ambition. An uncle of mine died when I was 20 of melanoma. And, ever since then, I want to, there's a couple of small kids. It was just so awful. And so I saw the end objective of molecular biology, the curing of cancer. And, but it was only in the second edition I talked about Warburg. I don't know, maybe the book was getting bigger or something, but, and then he disappeared from the third edition and appeared in the fourth. We were so dominated by genes. And then, the most striking fact is that, that in the book, I have 3 copies of it. I look at virtually every day, Bob Weinberg's book, Biology and Cancer. There's not in the reference one mention of Warburg, lactic acid, or glycolysis. So molecular biologists aren't as all-knowing as some of them think they are. So, and you can see that the end is just sort of bullshit. At the end, something's different with the membrane of the cell. I'm trying to explain it. But now, the Warburg effect, really you couldn't forget, because one way is just the PET scanning. You just go through and you see, is glycolysis high in the cancer, and if it is, then you have high lactate, and your patient's probably going to die pretty soon. And these facts have been around for 20 years, so it makes it more remarkable that the community just went on and just never wanted to explain Warburg. And now, finally, they're back to it. Luke Cantley and Craig Thompson, especially Craig, who's just written about, the biochemistry of cancer. And really very good news, he's the new head of Memorial Sloan Kettering. And, he not only wants to cure cancer, he wants to prevent it. He really realizes that, fundamentally, you start with genes, but you end up with metabolism. And the metabolism is changed, and, and you've got to have biochemists. Now we're training very few biochemists in the United States. Big mistake. Harvard even didn't appoint someone. In the college, they're fads. Anyways, now one realizes that the fact that Van Potter said, the main function of glycolysis is not to produce ATP, but it runs very fast because you've got to produce the building blocks, the nucleotides, the amino acids, and so on. And you do this by blocking the formation of pyruvate, and it doesn't even get down to the mitochondria. They found the last enzyme, pyruvate synthase, exists in 2 forms, in dividing cells and in cancer cells. It's the M2 form. In the M2 form, you can phosphotyrosine groups force it to go into a dimer which doesn't have any activity, and so when you turn on growth, you shut off glycolysis feeding the mitochondria. And this now is shown very nicely. So one of the hopes was maybe you can inhibit pyruvate kinase and so on. It has the usual problem. You also is there to divide every ordinary cell. So, but it's important. One person kept going. There was really only one person in the United States who tried to base his career on studying glycolysis. As Peterson at Johns Hopkins. And he was never quoted by anyone. He was just sort of ridiculed. He believes in Warburg. But Warburg was a hard fact. But what he actually showed was that in cancers, the hexokinase goes and actually sits in the pore, in the mitochondria. Very adaptive. Now, back to Folkman. What Judah wanted to do was to block angiogenesis. He realized, and by that stage it was clear that all cells require sort of signals to divide, and they come from growth factors. And that Judah's aim was to find the growth factors for endothelial cells. And if you found the growth factor, then you could inhibit them. Okay. And the real big step came from Napoleon Ferreira at Genentech, who found vascular endothelial growth factor, which other things will promote angiogenesis, but it seemed to be the big one. Okay. And once you had that, then you could you find an inhibitor of it, and could you prevent its binding to the receptor? Could you inhibit the receptor, etc. And so we began to get, the biochemists began to get the first inhibitors of the growth factors. And then you needed a system to study it, and then in the early 80s were developed the first genetically engineered mouse models of cancer. And Doug Hanahan, who I show here at Cold Spring Harbor when he came down from Harvard as a junior fellow, made one based on the which turned on the islet cells in the pancreas, called a RipTag model. Anyway, so it led to the expression of the mice normally developed islet cell carcinomas. And Judah began collaborating with Doug, so they published a series of papers together. And there was a lot of the islets became hyperplastic, some became, blood vessels grew in, a small percentage went on to being carcinomas. So Doug, with this system, began adding inhibitors. And here's Judah giving a talk at Cold Spring Harbor because they made a second big discovery. Besides, you could block angiogenesis by normal inhibitors of angiogenesis, which just exist in the blood serum. And they are all essentially formed by breakdown of proteins in the extracellular matrix, collagen breakdown, etc. The first one was a thrombospondin founded by Noel Bach. She found it at Northwestern in the early, about 1991. And then Judah found in his lab endostatin and that. So you had a sort of angiogenic. You had pro-angiogenic growth factors, and then you had natural inhibitors. And what determined the balance? And in the transgenic thing, you could then begin to add and show that the anti-angiogenic proteins actually worked. They never stopped completely the tumors from growing.